In 1996, a soil scientist named Sara Wright at the United States Department of Agriculture was studying a routine soil sample when she found something that should not have been there. A protein, sticky, abundant, and completely unidentified in any previous literature. She named it Glomalin after the Glomal's fungi that produced it. Within 2 years, her team discovered that this single compound accounted for up to 30% of the total carbon stored in soil. 30%. One substance produced by one class of organism holding together a third of the carbon beneath your feet. Her findings meant that the living infrastructure responsible for soil fertility was not a chemical equation. It was biological. It was fungal. And it had been quietly running beneath every healthy ecosystem on the planet for over 400 million years. The cost of rebuilding it in degraded farmland? A few dollars worth of fungal inoculant and 1 season of patience. The ancient Romans understood something was alive in their soil. Pliny the Elder documented the practice of transferring earth from productive fields to barren ones, specifically to carry the invisible vitality that made crops grow. Japanese rice farmers for centuries returned organic residues to their paddies, unwittingly feeding the fungal networks that kept their yields stable without synthetic input. Indigenous peoples across the Amazon created terra preta, black earth so biologically rich that soils amended over 1,000 years ago still outperform surrounding jungle soil today. And the global fertilizer industry, now worth over $200 billion a year, has spent the better part of a century making sure you never learned any of this. Today, we are opening a vault that connects a 400-million-year-old biological network to a USDA laboratory discovery to the dead patch of garden in your backyard. And by the end of this video, you will know exactly how to restore your soil permanently without buying another bag of synthetic fertilizer again. The story begins not thousands, but hundreds of millions of years ago, in the Ordovician period, when the first plants began crawling from water onto bare rock. They had no roots, not as we understand them. What they had were partners. Fossilized specimens from the Rhynie Chert in Scotland, dating to roughly 410 million years ago, even show fungal hyphae threaded through the earliest land plant tissues. This was not parasitism. This was architecture. The fungi dissolved minerals from rock and delivered phosphorus and nitrogen to the plant. The plant, in return, fed the fungus sugars produced through photosynthesis. This exchange, mycorrhiza, from the Greek myco for fungus and rhiza for root, is the oldest known mutualism in the terrestrial fossil record. Plants did not colonize land alone. They were carried there on a fungal scaffold. Fast forward to human civilization, and you find this partnership recognized, if not fully understood, on every inhabited continent. In Mesoamerica, the Milpa system, the Three Sisters planting of maize, beans, and squash, created polycultures that naturally supported diverse mycorrhizal communities. The roots of each crop fed different fungal species, which in turn knitted the soil into a self-sustaining web. Yields remained stable for centuries without any external fertilizer. In China, rice-fish culture systems dating back over 2,000 years maintained paddy fertility through biological cycling. Organic, matter-fed soil fungi that locked nutrients in place, preventing the leaching that plagues modern monoculture. In West Africa, traditional agroforestry systems intercropped food crops beneath nitrogen-fixing trees, creating layered root zones colonized by overlapping mycorrhizal networks that outperformed adjacent plots treated with synthetic fertilizer year after year, without any purchased input. And then there is Terra Preta do Indio, the black earth of the Amazon. Created by indigenous peoples through deliberate addition of charcoal, bone, and organic waste, these soils are self-renewing. Plots last managed over 1,000 years ago still outperform surrounding jungle soil. The charcoal provided a porous habitat for mycorrhizal fungi and beneficial bacteria, creating a living matrix that regenerates itself. This is not folklore. This is convergent biology. The same fungal truth discovered independently by civilizations separated by oceans, millennia, and language. Every one of them found that if you feed the soil's living network, it feeds you back. Permanently. The mechanism is elegant and, once you understand it, impossible to unsee. Mycorrhizal fungi form 2 primary associations with plant roots. Arbuscular mycorrhizal fungi, AMF, penetrate root cells of roughly 80% of all terrestrial plant species, creating branching structures called arbuscules that serve as exchange sites. Phosphorus and micronutrients flow from fungus to plant, carbon sugars from plant to fungus. Ectomycorrhizal fungi wrap around root tips in a dense sheath and extend vast, high-fall networks outward. These are the fungi of forest trees. Here is the critical number. A single root can access 1 to 2% of the soil volume within its reach, but mycorrhizal hyphae extend that reach by a factor of 100 to 1,000. 1 cubic centimeter of healthy soil can contain 200 meters of high-fall filament. The fungus does not supplement the root system. It replaces its limitations entirely. And glomalin, the compound Wright discovered, is the glue holding it all together. This glycoprotein persists in soil for 7 to 42 years after the fungus dies. It binds soil particles into stable aggregates, creating the crumb structure that holds water, resists erosion, and stores carbon. Without it, soil compacts and washes away. With it, soil breathes. This is biological infrastructure, not a chemical input. That distinction is enormous. Synthetic fertilizer delivers nutrients that wash out with the next rain, leaving soil no better and often worse because high concentrations of soluble phosphorus actively suppress mycorrhizal colonization. The fungi detect the flood of free nutrients and withdraw. Why build an expensive network when phosphorus is being handed to the plant? The plant becomes dependent. The soil biology collapses, and the farmer buys more fertilizer. Let that sink in for a moment. The product designed to feed your soil is chemically configured to destroy the one organism that feeds your soil for free. The modern validation of mycorrhizal science reads like a quiet revolution that nobody was supposed to notice. In 1997, Suzanne Simard at the University of British Columbia published a landmark paper in Nature demonstrating that Douglas fir and paper birch trees were exchanging carbon through shared mycorrhizal networks. She used radioactive carbon isotopes to trace the flow and proved that trees were not competing in isolation. They were cooperating through fungi. Older trees subsidized younger ones. Dying trees dumped their carbon reserves into the network for neighbors to absorb. The forest was not a collection of individuals. It was a superorganism connected by fungal infrastructure. Her work has since been replicated worldwide. We now know that a single fungal network can connect hundreds of trees across multiple species, preferentially routing nutrients to stressed individuals. In agricultural contexts, the data is equally striking. A 20-year field trial at the Rodale Institute in Pennsylvania compared organic systems relying on biological soil management against conventional synthetic approaches. The organic plots, rich with mycorrhizal fungi, matched conventional yields for most crops and dramatically outperformed them during drought years because the fungal networks improved water retention by up to 30%. The organic soils also sequestered significantly more carbon, turning farmland from a carbon source into a carbon sink. At the University of Zurich, Marcel van der Heijden demonstrated that plants colonized by mycorrhizal fungi absorbed up to 80% more phosphorus than non-colonized controls. When mycorrhizal diversity increased, multiple fungal species rather than one, plant productivity climbed further because different fungi specialized in different nutrients. The evidence is not anecdotal. It is institutional, peer-reviewed, and reproducible. The fungal network works. So, if the science is this clear and this well-documented, why are you not learning about mycorrhizal inoculation in every gardening guide, every farming manual, every agriculture class? The answer is not a conspiracy. It is something more ordinary and honestly more frustrating: economics. You cannot patent a fungus that has existed for 400 million years. You cannot patent glomalin. You cannot patent the symbiotic exchange between an arbuscule and a root cell. And what you cannot patent, you cannot monopolize. What you cannot monopolize, you cannot scale into a quarterly earnings report. The global synthetic fertilizer market generates over $200 billion in annual revenue. The three largest producers, Nutrien, Yara International, and the Mosaic Company, are publicly traded corporations with shareholders expecting growth. Their business model depends on one assumption: that the farmer needs to come back every season and buy more product. A mycorrhizal network that rebuilds itself, that persists across years, that transfers nutrients for free, that is not a product. That is an existential threat to the model. You cannot build a subscription business around a fungus that pays for itself. You cannot justify a $60 bag of granular fertilizer when the active biological alternative costs $4 and lasts indefinitely. And you certainly cannot maintain a $200 billion industry when the solution is already in the soil, waiting to be reactivated. So, the knowledge does not get suppressed through any deliberate conspiracy. It gets suppressed through something more efficient: irrelevance. It is simply never promoted. Search for how to improve your garden soil online. The first page of results will be dominated by paid advertisements for synthetic fertilizer brands, sponsored content from chemical companies, and retail links to commercial soil amendments. Mycorrhizal inoculation, if it appears at all, will be buried on page 3 or 4 behind a wall of commerce. Not because anyone censored it, because no one with a marketing budget has any reason to promote it. Walk into a garden center. The fertilizer aisle is enormous. Brightly colored bags with promises of instant green, fast growth, maximum yield. The mycorrhizal products, if the store carries them at all, occupy a single shelf in muted packaging. No brand ambassador, no television commercial, no influencer partnership. Open a standard high school biology textbook. You will find extensive sections on plant nutrition, soil chemistry, and the nitrogen cycle. You will find almost nothing on mycorrhizal symbiosis. The curriculum was not designed to suppress this knowledge. It was designed by people who were never taught it themselves, because the research funding that shapes academic priorities has for decades flowed toward patentable chemical solutions, not toward a fungus that cannot be owned. Even the language has been reframed. We say plants need to be fed. We say soil needs to be amended. We speak of deficiencies and corrections, as if the earth is a patient and the fertilizer is medicine. The framing itself makes buying feel like the only responsible action. Nobody says the soil needs to be reconnected, because reconnection is free and you cannot sell free. Now, in fairness, synthetic fertilizers are not entirely without merit. In acutely depleted soils, an initial nutrient boost can help establish plants while biological systems rebuild. In large-scale commercial agriculture operating under extreme time and cost pressures, the predictability of synthetic inputs has genuine logistical value, and certain high-demand crops in specific climatic conditions may benefit from supplemental feeding even in biologically healthy soil. But for the vast majority of home gardens, small farms, landscapes, food forests, and restoration projects, which account for hundreds of millions of growers worldwide, the mycorrhizal approach is not just cheaper. It is self-sustaining, it is permanent, and it has been proven effective for 400 million years. Here is exactly how to do it. First, understand what you are buying. Commercial mycorrhizal inoculants contain spores of arbuscular mycorrhizal fungi, ectomycorrhizal fungi, or both. For vegetable gardens, flower beds, and most food crops, you want an AMF-dominant blend. Look for species like Rhizophagus irregularis, formerly known as Glomus intraradices, on the label. For trees and shrubs, especially oaks, pines, willows, and birches, you want an ectomycorrhizal blend. Many quality products contain both. A 1-pound bag of granular mycorrhizal inoculant costs between $4 and $12 and will treat an entire standard garden bed. Application Method 1. Direct root contact at planting. When transplanting seedlings or starts, sprinkle roughly 1 teaspoon of granular inoculant directly into the planting hole so it contacts the roots. The spores must physically touch root tissue to colonize. This is the most effective method. Push the roots gently into the inoculant, backfill with soil, and water normally. Near Application Method 2. Seed treatment. For direct sown seeds, mix your seeds with a small amount of inoculant in a bag and shake gently until the seeds are coated. Plant as usual. As roots emerge, they will immediately encounter spores. Application Method 3. Existing plants. For established gardens, mix 2 tablespoons of inoculant per gallon of non-chlorinated water. Chlorine kills fungal spores. If your tap water is chlorinated, fill a bucket and let it sit uncovered for 24 hours to off-gas, or use collected rainwater. Water the solution directly at the base of each plant, soaking the root zone. Repeat once a month for 3 months to establish colonization. Critical rules for success. Stop using synthetic phosphorus fertilizers. High phosphorus inputs are the single biggest killer of mycorrhizal colonization. If you must fertilize during the transition, use low-phosphorus organic options, compost, worm castings, diluted fish emulsion. Stop tilling deeply. Tilling shreds high-fall networks. Use shallow cultivation or no-till methods. Keep living roots in the soil year-round by planting cover crops in the off-season: clover, rye, vetch. Living roots feed the fungi. Dead, bare soil starves them. Mulch the surface with 2 to 4 inches of organic material. Wood chips, straw, shredded leaves. This moderate soil temperature retains moisture and provides a slow feed of organic matter that supports the entire soil food web. Within one growing season, roughly 3 to 6 months, you should observe darker, richer soil color, improved water infiltration, and stronger plant growth with less watering. Within 2 to 3 seasons, the mycorrhizal network becomes self-sustaining. It will recruit new fungal species, connect new plants, and expand its reach without any further input from you. The total cost: $4 to $12 for inoculant, a bag of cover crop seed for under $10, and free mulch materials from your own yard or local tree service. Compare that to $100 to $300 per year in synthetic fertilizers, soil amendments, and the water bills that come from soil that cannot hold moisture because its biological structure has been destroyed. That dead garden bed, the one with the gray compacted soil that cracks in summer and floods in spring, it does not need more chemicals. It needs its fungal network back. 400 million years ago, the first plants that left the ocean did not do it alone. They walked onto bare rock on a bridge built by fungi. The peoples of the Amazon knew it. Mesoamerican farmers knew it. Chinese rice cultivators knew it. Your great-grandparents composting kitchen scraps into their garden knew it without a single scientific paper to confirm what their hands could feel in the soil. The earth does not need to be fed. It needs to be reconnected. And reconnection cannot be sold, bottled, branded, or marked up. Which is exactly why no one with a profit motive will ever tell you this. Every share puts a crack in the wall built around knowledge that was never anyone's to lock away. If this changed how you see the ground beneath your feet, subscribe and hit the bell so you never miss what we uncover next. Every like tells the algorithm this matters more than another ad. Stay curious. Stay rooted. I will see you in the next one.